Hello, our topic is ethical dilemmas on CRISPR. In our group, we have Luke Solis, Ryan Farmer, Ryan Fercucci, and Isabella Daniels. These are our learning objectives. In this presentation, we will be going over what is CRISPR, the ethical dilemmas with CRISPR, the risks involving CRISPR, and the benefits. So what exactly is CRISPR? The acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is known to be a revolutionary gene editing tool. The types of changes that occur in DNA sequencing include disruption, deletion, and insertion. By utilizing the Cas9 enzyme and guide RNA, CRISPR enables scientists to target specific DNA sequences within the genome. The Cas9 enzyme cuts the DNA at the targeted location, initiating DNA repair process that can lead to specific modifications or alterations in the genetic information. This ability to precisely cut DNA and manipulate repair process is what makes CRISPR a powerful tool for gene editing and genome engineering. Disruption occurs when the Cas9 enzyme guided by the gRNA cuts the target DNA sequence, leading to a loss of gene function or inactivation. Deletion can occur when the repair machinery inaccurately joins the cut ends, resulting in the removal of a fragment of DNA. Insertion is achieved by providing a template DNA sequence along with the CRISPR components, allowing for the precise incorporation of new genetic material as a targeted site. These processes of distribution, deletion, and insertion are key mechanisms by which CRISPR achieves precise gene editing and manipulation of the genome.
Hello everyone. I will now discuss two important ethical dilemmas of CRISPR. The first being designer babies and the controversy around this topic, and then we will talk about the inequality and access to CRISPR. So what are designer babies? These are babies who have chosen and altered genetics to better suit an individual's desires in a child, which could range from changing one's appearance to changing the code for a disability or desired skill. So what ethical dilemmas can arise from this tool to create the perfect child? The first being, the children whose genes are being altered are not able to give permission. As an unborn child, they cannot reject the idea of changing their natural appearance or abilities, when it is themselves being most affected by the change. The second being, other than for disease, why is there a need to change an individual's natural DNA? We should be learning to embrace the natural genetics we inherit, rather than try to alter one's child into an expectation. With that being said, is there a need for regulation? With this desire to choose an individual's genetic makeup, how far will parents go? Do we as society have a say in what others choose to do for their children? This brings us to the topic of inequality and access to CRISPR. CRISPR costs $1 to $2 million to use, and it is only available in a small number of medical facilities worldwide. This meaning the tool is very inaccessible to all parties, but rather favors those more wealthy and able to make the distance. This meaning the upper class in urban areas have an advantage over others, which could also lead to a higher rise in inequality in society. Having the ability to choose or alter an individual's code is a privilege that can create tension between those who need the benefits and those who use the tool for their own benefit, which is not fair for society to divide between due to the prices and lack of locations. I will now pass it on to talk about the risks and benefits of gene editing.
Now we will discuss the risks associated with CRISPR. Despite the precision of CRISPR, there is a risk that the gene editing process may result in unintended changes in the DNA sequence at locations other than the intended target. These off-target mutations can introduce new genetic variations or disrupt the normal functioning of other genes. CRISPR can be potentially harmful to the human embryos due to the risk of unintended off-target mutations and other unforeseen genetic alterations that can have long-term consequences. Another risk includes the long-term effects of CRISPR on future generations that refer to the potential impact that gene editing in one generation may have on subsequent generations. This is particularly relevant when considering germline editing, which involves making heritable changes to the human genome. Germline editing raises concerns about the long-term effects on future generations, and also our last risk, the potential for altering the human gene pool. The potential altering of human gene pool raises concerns about the possibility of natural genetic variety being lost and the introduction of artificial or designer features into the human population. The concept of altering the gene pool by deliberate genetic mutations raises worries about the possibility of unanticipated consequences, as well as the ethical issues of permanently altering humanity's genetic history.
As you have heard, CRISPR has the potential to correct genetic diseases by modifying the mutations that are the actual causes for the disease. Hereditary diseases like hemophilia, Parkinson's, and Tay-Sachs are just a few of the disorders that CRISPR could be used to alleviate. The potential to accurately and swiftly treat or prevent the diseases that have control over people's lives is only one of the seemingly endless benefits of the utilization of CRISPR, and it is why it has the biomedical community so interested. While these potential benefits to humans are incredibly interesting and have immense potential, it is worth noting that CRISPR could benefit people even if human DNA is never altered. CRISPR could have the ability to also alter the genetics of other organisms, like an endangered species, to make it embody more desirable traits that will boost their ability to adapt, survive, and reproduce. It could also prevent a species like mosquitoes, which are responsible for transmitting diseases like malaria or West Nile virus, from being able to do so anymore. The last benefit of CRISPR that I will be talking about today is its potential ability to alter the genetic makeup of plants or crops, to make them even bigger, extra resistant to pests or diseases, and even possibly more nutritious for humans. This could prevent humans from running out of food and make them live overall healthier lives. But while these possibilities are beneficial for the human race, it is important to think about the ethical issues behind genetically altering an organism, population, or its environment.
Now that we have learned about CRISPR, let's summarize the key points. The main ethical issue associated with this gene editing tool are concepts of designer babies and the inequality and access of CRISPR. The risks associated with CRISPR include off-target mutations, long-term effects on future generations, and potential human gene pool altering. Lastly, CRISPR can treat genetic disorders, it can create genetically edited organisms, and make agriculture. And now here is a list of the resources we used. This includes our presentation on the ethical dilemmas associated with CRISPR. Thank you for listening.